So I've been locked away in my house for a year and a half, and I came in today. I forgot a shirt, so I've worn a polo shirt. So you're just going to have to imagine I'm Californian for the day, and it'll seem normal. But yeah, Toby, Hampshire. So I'm a senior manager for clinical scientific affairs at the microbiology department for Thermo Fisher Scientific, and I've also invited John along, John McCarthy. I'm going to waffle on for 10 minutes and tell you a few things about blood culture processing and what we've been looking at as part of my role in the business, and link that to a product plug, to be obvious, but I'll keep that to one slide. But we were also asked, can we bring some case studies along? And obviously, if I'm doing a commercial talk, I don't have access to case studies like some of these gentlemen at the front. But we've been working for about a year and a half with the Sepsis Trust. So John is a sepsis survivor. So we wanted to dovetail what we were presenting to you today, make sure that John also gives a real life case study from his perspective. So that's what we'll aim to do over the next 20 minutes. And I'm going to take my watch off and keep an eye on myself, because I'm known for talking for way longer than I'm supposed to. Apologies. So, marking it, I'll stop at 1:30. So, as I said, I'm going to start talking to you. I'm going to start talking to you about blood cultures and how they're processed in the UK. Everything written on the next couple of slides is actually just extracted from what was the Public Health England's UK standards for microbiology investigations, essentially for blood cultures. Obviously acknowledging the fact that there's lots of fantastic organisations that work with Public Health England in this case to create those standards. I think it was last updated in 2019, so you see on some of the screenshots it's published on the web. Nice, that's the great thing about this country, I think it's freely available. But it's got under review all over it because as a watermark, because I guess it's being looked at again. So here's some statements I just pulled out of that. And clearly investigation of infection of the bloodstream is of significant importance. And the commentary in these guidelines alludes to that. So antibiotic resistance among pathogens, particularly gram-negative pathogens that people struggle with the most, is the most actually frequent cause of ineffective empirical treatment of bloodstream infections. It's really important for early identification and susceptibility testing. And actually that helps a lot in terms of the time frame. It reduces morbidity, mortality, and improves patient care. The guide talks extensively all the way through it about decreasing turnaround times and how important that is. And it looks at the different stages through the process, because we often forget, I'm originally a long time ago from a lab as well, and you tend to just think about things as, well, what's done in the lab. But obviously, if someone takes a blood culture, there's all the work that's done before, and where does the blood culture end up? Where does the blood bottle end up? Does it end up in that hospital? Does it need to be transported by a courier? So there's all kinds of other things that you have to take into consideration. The top right, I kind of cut a bit off, but essentially it talks as well about reducing the time between sampling and reporting results as much as possible. And it also goes on to say, well, you should, as an institute, also break down the process and study it and understand it, and where you can reduce those times. Essentially it's saying, although there's an admission that there's some publication on this, it's not definitive data. There's some robust data lacking, and one of the things I'll be doing in the next couple of years is working with my organisation and also with other organisations to try and fill some of those gaps, actually. Also, the final point on this slide is it asked institutes to regularly audit what they're doing and consider whether improvements can be made. So then last slide on the blood cultures, because it's quite dry, I know, but it's important that the guide goes on to talk about pre-analytical, analytical and post-analytical standards, and they're actually specified, so I highlight them in yellow. So from collection to incubation, they're saying that it really, well, the recommendation is that should be in less than 4 hours. Gram stain in less than 2 hours. Sensitivity results is a little bit more flexible because it takes time or it did take time to get definitive sensitivity results. So there's 24 hours given. And key also, okay, you get a result, you get an important result in terms of whether the patient might be on the right or wrong antibiotics. So it should be reported within 2 hours. And actually, we've got some I know because I've been going around a couple of European sites and a UK site recently, there's more and more hospitals actually connecting the limb system in the microbiology department to the bedside, and more microbiologists are getting involved with actually reporting results that appear at the bedside immediately. And that's got to be a positive thing, and I think it's a trend that will carry on. So I mentioned I was a clinical scientific affairs manager within Thermo. What that really means within my microbiology department is that half the business doesn't actually know what I do, but let's move on from that. So here's an example where I sat with some people in different parts of the business, and we just played with the idea of how you would manage someone with a severe bacterial infection within the hospital based on what we can provide. And there's PCT, so there's biomarker testing. Actually, it's a really good example. I think people think of PCT as something, as a test that you can define whether someone's got bacterial viral infection, and it's done once at the start. But there's actually loads of really good examples now where hospitals are testing on a 12 or 24-hour basis, and you can use it to look at reaction to antibiotics and also improvement of care. The gram stain is the sort of central microbiology part to this. Then you see something on there called ASTAR or A-star, depending on how you want to pronounce it. That's what I'll tell you a little bit about. So it's a sensitivity testing device now that will deliver sensitivities in about 6 hours, which is significantly quicker. And I don't mind saying there's other devices on the market. Sensitizer is a long-term product that we produce that helps people in terms of dosing strategies, when dosing strategies come in, particularly for critically ill patients. And longer term also, there's this interest in therapeutic drug monitoring and measuring the amount of antibiotics in someone's blood so that you can keep a more consistent level at the site of infection. You don't need to see the detail in this one, but it's just a really good example of, we also independently tried to study the blood culture process, and the top part of this slide is actually work we did internally to look at where there might be awkward delays that we'd rather remove from the process, but are always there. Classic examples, as I talk to people, as I said, in hospital throughout the UK, talk to Ron Daniels, I've been working with in the Sepsis Trust. It's clear that blood cultures are sometimes taken and not processed immediately. It all depends on what kind of care the individual hospitals can offer, and unless there's a 24/7 coverage, sometimes they get taken, but maybe not get processed. In the most, I guess, in the most difficult situations, you might have someone where blood cultures are taken late on a Friday and they might not be processed till Monday. But I think that's reducing. I think things are improving. The bottom half of this slide is actually, again, screenshots from the recommendations by Department of Health. Well, they've done exactly the same thing, actually. On the bottom left, you can see this sort of area where they've highlighted where delays can happen. Bottom right is talking about the therapeutic window. So you can see those 4 graphs, and essentially they're different scenarios, whether someone's putting on appropriate treatment immediately, or whether they're put on an inappropriate treatment. If you're putting an inappropriate treatment, you have a therapeutic window for that individual patient, and only if you put them on the right antibiotic within that therapeutic window is their outcome likely to improve. And so really what we're aiming for in worst case scenario is that bottom right graph where you've got this therapeutic window. It's obviously highly important that it's linked to time, but if you're on inappropriate treatment and you get the right information quick enough to the bedside, then you can change and you can put appropriate treatment and hopefully improve outcomes. So why am I telling you that? Here's the sales plug, sorry. So actually it was the timing is good. Last Thursday we launched this instrument along with our partner Q-Linear. It's an instrument that would sit in the microbiology lab. It doesn't have to sit in a microbiology lab because we've already had feedback. So we did 2 UK evaluations with this in 2 UK hospitals. Feedback was actually it's so straightforward to use that there was an idea that there's situations, if you like, where there's satellite hospitals that courier blood cultures to an alternative hospital to get them processed. Actually, you just draw 2 ml out of 2 ml of blood out of the bottle and add it to a cartridge, and you add the cartridge and the disc to the instrument, and it runs in 6 hours and delivers the results. And with interpretation, I've got to speed up because I'm running out of time. So part of the reason I'm also presenting today is just to highlight that there will be part of the business I work for now that goes for a sales side, and they'll do demos of this instrument and look at it from that point. My role is actually to look at it from a health economic point of view, and so I'm working with the manufacturer Q-Linear on areas that we think are important, like the sample setup time. And we're trying to identify accounts across Europe that essentially have an interest in how can we improve blood culture processing, how can we speed it up, and not just at the point of getting a sensitivity test, although that is a bottleneck, but also looking at the whole process and working as a group to do that. Likewise, it's a broad panel, so there's more the traditional approach to a lot of the tests that you see on the market. Look at about 100 different antibiotic solutions at a time. This thing looks at about 300, so it's about 3 times as much data. Very useful for therapeutic drug monitoring and PKPD based therapeutic drug monitoring, I should say. And so we're essentially interested in identifying NHS hospitals that might want to put one of these in play with this as a potential and a system to improve what they're doing. And likewise, here's the final one. So we didn't initially consider it, but we recognise actually, if you look on the left, so Q-Linear is a company we're working with. We don't just process aerobic bottles. It's validated to process with anaerobic blood bottles, and it's actually works with paediatric blood bottles. So again, we're already actually in talks with a paediatric hospital with a large paediatric population in the south of the UK about putting one of these systems in again so that we can improve it from that perspective. Anaerobic bottles quite often flag up earlier than aerobic bottles, so it's an advantage that the system does both. But essentially, that's my future for the next year or two is working with some organisations, and actually the business we're in, plus Q-Linear, who we're partnering with to put this instrument out there, investing in health economic studies. There's going to be some in Italy, there's some in Northern Europe, and we're looking probably to put some more in UK, France and Germany. So having said all of that, I said I didn't have a case study. I invited John because all of this consultation process we've been through as a business and working to try and understand why we're developing this device, and also educating people within our business that didn't know anything about sepsis. We partnered with the Sepsis Trust, and John's a sepsis survivor, and I just invited him here today for you to get sort of first-hand example of someone that's been through the care pathway with sepsis, and perhaps some of the pitfalls as well as the successes, and it's a positive story.
I'll let you be the judge of whether it's a positive story or not. Thank you, Toby, and thank you to Thermo Fisher for giving myself and the UK Sepsis Trust an opportunity to come and speak to you today. I am not medically trained. This will become very apparent throughout the talk. Most of that goes right over my head. I do try, but I don't understand it, to be honest. 5 years ago, Cheltenham Racecourse. I'm originally from Liverpool, but I'm posh now. I live in Stratford-upon-Avon. Every year, all my family, Liverpool, Isle of Man, Europe, we all descend in Stratford. The posh ones stay in the hotel. The poorer ones are on my floor. We get what we call a charabanc. There's about 50 of us. We go to Cheltenham races every year. It's Christmas for adults. No different this year. Now, I'm sure you've all been to a concert, a gig, or a festival, and it can be really busy. You're trying to get from one end of a place. People are coming towards you. I'm in the Guinness village. I'm trying to get through. I take a step too far to my right-hand side. Bang my leg. Ouch. It hurts. I know it's going to be sore in the morning. Family say to you, you've got bruising on the bone there, lad. I don't know if that's a medical term or not. Carried on with the day, obviously, as the day wore on. I became more and more anaesthetised, and the hobbling stopped. So the next day it was sore. It was really sore. I was thinking, oh, God, just got to be painful for a few days. Then the next day it was starting to redden, and I was feeling awful. I wasn't feeling good at all. And I'm starting to think to myself, oh, God, I'm getting old. These hangovers are taking longer to get rid of. And I just felt myself getting worse and worse throughout the day. And I just knew something wasn't right. I woke up the next day. I was feeling a bit confused. I let it go. And in the end, I said to my brother, can you take me to the hospital? So my brother and my friend come round, because instinctively and intuitively, I knew something wasn't right. I knew there was something really wrong with me. So I goes to the hospital. I'm in A&E. I'm going to tell you the hospital. It was Warwick Hospital. I'm waiting for about half an hour. Not so bad. It goes through. Doctor sees me, x-rays, poking, prodding, all that sort of stuff. At the end, he says, Mr. McCarthy, you have cellulitis. I said, okay. He said, there's your fluoxetine. Go see your doctor, your GP, in 7 days' time. Okay. No blood's taken. No questions asked. Nothing like that. I goes back and I'm feeling embarrassed going back in the car, because I packed an overnight bag, and I'm saying, how daft am I? Because I just knew. The following day I'm feeling even worse. I'm getting more and more confused. The skin's becoming mottled. Family and friends around there saying, go back to the hospital. I'm saying, no, I've got to listen to what the doctor said. I've got to let these antibiotics work. Following morning, I noticed my bed really damp where my leg was, where I banged it. My leg was seeping. I felt if I was going to put weight on the leg, it was going to snap. I was breathless. I was feeling like I was going to die. So again, the same people took me back to the same hospital. I'm in A&E. I had to get in a wheelchair to go into A&E. I was sat there for 30 seconds, and a puddle was starting to form at my foot, and a lady come over. To this day, I don't know if she was a patient, a doctor, I don't know, but she got me into A&E, and into triage, resus straight away. Same doctor, markedly different response this time. I remember getting strapped to a gurney and the cannulas and the drips and everything. And it was 3.5 months later I left that hospital with my right leg amputated above the knee, having spent 17 nights in ITU. My family had all been with me, and all my friends who'd been with me at Cheltenham, they'd all obviously gone home. They all had to come back. They're getting told, he's not going to make it through the night. On 2 occasions, fortunately, and obviously, I did. And people forget, I think. They think about the individual, but they don't realise the stresses and strains this puts on family and friends, particularly when it's an unknown disease, or they couldn't get their head around it. They weren't told what it was either. Nobody explained what it was. When it finally come to me to be leaving the hospital, basically I was just told, there you go, crack on now, get on with your disabled life. The flat that I bought, I couldn't go back to. It was not disabled friendly. So they found me a flat that I had to go into, and I had to learn how to use a bathroom with one leg, how to get around. And initially I was euphoric. I was thinking, yeah, hey, I'm the boy who lived. Anybody who was asking me, flesh-eating bug got in my leg. So I was telling everyone, because that's what somebody told me. Nothing to do with sepsis. No mention of sepsis. It was only 6 to 8 months later when I was suffering from depression. I was having the most intense nightmares. I was so fatigued, I was pushing people away. My brother finally got in touch with me, and he said, let's go through these drugs that you're taking. Because all that was happening, I'm going to see my GP. Have that antidepressant. I'd go back 2 weeks later. Oh, no, about that one. You have that one. And I come through a list of everything. It was gabapentin. Never forget it. And it said treatment for amputees and for nerve endings, and it mentioned sepsis. So sepsis is that not what I had? So I Googled sepsis then, and that led me to the Sepsis Trust, and it led me to realise that I was not unique, that this happens all the time. This happens around the country, and it kills over 60,000 people a year, and there's 250,000 people a year just in this country who lose limbs. May only be the toes, but it may be all 4 limbs and the nose. And I couldn't believe it. I went to see the Sepsis Trust, and then I found out about post-sepsis syndrome, which was what I was struggling with, and that includes PTSD, because your brain can't compute where it's at when you're spending so much time in ITU. It comes up with these horrendous themes of murder and kidnap. No one told me. No one told me that. No one told me about the fatigue, the sleeping 18 hours a day, the cognitive ability that will be impaired for the rest of my life, and I'd never be able to go back to work to the standard that I was at before. I was able to go into the doctors and really front them and have a go and say, what are you doing? You must have other people, elderly and people who've got kids, and you're just giving them antidepressants. And they all went, we don't know anything about it. Long COVID's in now, everyone knows about that. It's just the same as post-sepsis syndrome. Now, the thing is with sepsis is that there are 6 denominators. The S is slurred speech or confusion. I had that. Extreme shivering, muscle pain. Oh, yeah. Passing no urine. This is easy, this one. All you have to do is ask someone, have you been in the toilet? I can't remember if I had or I hadn't. Severe breathlessness, it's your throat constricting. I had that. Feeling like you're going to die. It's very difficult to pinpoint this, but everybody who's gone through what I've gone through says the same thing. It's an aura. You're going to die. And severe mottled or discoloured skin. I had all that. That was my leg. All 6 signs. I'm presenting at Warwick Hospital, and they give me floxacillin and sent me away. All it took is one person within my big circle of friends and family to have knew what sepsis was and said, hey, John, why don't you ask her to be sepsis? And I wouldn't be here. And if the hospital had thought, because everything was pointing to this, if they'd have followed the sepsis 6, which is buffalo, take the bloods, test it. They didn't do it. Test the urine, fluids, fill them for fluids, the fluids are going. Now, I know we talk about microbial resistance, but if you detect sepsis, then give them the broad spectrum antibiotics, because otherwise they're going to die, or otherwise it's going to cost you an absolute fortune because they're going to lose limbs. Go and open that big cupboard of antibiotics. Go and do it if you're suspected sepsis. This is the sort of thing you should be doing. Lactate, the lactate machines in hospital. Check the lactate, give the patients oxygen. Do you know how long that takes in hospital? 10 minutes. Do you know how much it costs? 90 quid. Do you know how much it costs if you don't do it? And people lose legs and whatever. Can you imagine the cost and the ongoing cost to me? Particularly when I sued them because of the negligence. And it was all unnecessary. All unnecessary because they didn't follow their own procedures. And they didn't take the bloods. And they gave a snap judgment and gave me the wrong antibiotics. But now I'm out talking 3-4 times a week. Well, I was before COVID, and I'm back doing it again. Raising awareness of sepsis and talking to you all and telling you the signs of it. So you can go back to your families and you can tell your friends, irrespective of whether you're a medical professional or not, because sepsis does not discriminate. It doesn't care how old you are. And honestly, you do not want this visiting your door. I'm really pleased with what Thermo Fisher and other companies are doing to spot the early signs of sepsis. But unless it's at the forefront of medical professionals' thoughts, and unless it's in the public domain and people are thinking about it, then we're never going to move any more forward. All I leave you with is just ask, could it be sepsis? Thank you for listening.